Welcome to Genomics Lite. This is What is a Gene? It's our first session of the year, and we've got Zachary joining us from the Wellcome Sanger Institute, who'll be talking to you this afternoon, this evening. And as we saw with the poll, actually the majority of people haven't been on a Genomics Lite before. So I will quickly go through a little bit about what Genomics Lite is. It's basically a series of webinars that we host from different teams across the Wellcome Genome campus. And I'll tell you a little bit about where we are based if you haven't heard of us before, a little bit later. But yeah, so we've got a whole series of webinars like this one that happen throughout the year. So do stay tuned if this is your first session for others that will be happening throughout the year. Just in case you haven't been, basically these events aim to help everyone to explore the science of genomics and what it means for our lives. We'll be throwing up different questions, as I said at the start, different polls and all sorts, and there'll be an opportunity for you to ask your own questions. So we just respectfully ask that everybody participate in the spirit of curiosity, and there might be some difference of opinions and views. We just ask that you will respect all of those different opinions and views if they occur. I will be moderating this session today. And I'll go on to a little bit. If you've never been on a Zoom webinar before, you'll see that there should be 3 buttons for you at the bottom. So there's a chat function, a closed caption function, and a Q&A button. If you have any technical issues throughout the session, just message me, use that chat button. I'll hopefully be able to sort those out for you, and I'll be able to, if you're not hearing the sound, something's not quite right, let me know and I'll see what I can do during this session. There's a closed caption button, so if anyone would like subtitles for this event today, if you just click that button, you will be able to have a live caption that will go alongside this talk today. And when we get to the Q&A bit, please do put all of your questions in the Q&A button. What I say is, if a question comes up and you think of it during the talk, do just put it in that Q&A bit, and we can get to it at the end. Don't hold on to your questions, essentially, just in case you forget them. Make sure you bung them into the Q&A when they come to mind, and then we can get to those straight away. So this is where we're based. So if you haven't heard of us before, we are based at the Wellcome Genome campus. We're in South Cambridgeshire. My name is Sam. I'm a science engagement officer. So my role is to basically do events like these. It's to share and create opportunities to share the work that goes on this campus. And we're joined by Zachary today to be able to go through a really interesting topic, which is essentially what is a gene. So what we'll be doing is Zachary will speak a little bit about this area. We'll have chance for a Q&A, and we'll be wrapped up around about half 5. This session is being recorded, so if for any reason you need to your connection drops or you need to nip off, there will be a recording, and this will be available on our website and YouTube to watch as well. So I will stop sharing at this point and hand over to Zachary for the start of the session. So let me stop sharing and hand over to you, Zachary.
Hello, everyone. I'm glad to be here. Sorry, I just swapped something. Okay, great. Sam, this is all working fine?
Yes. That's all coming out all clear on my end.
Okay, well, yes, hi everyone. This is a really basic topic in biology. What is a gene? But the more we think about it, the more we can learn about biology. And actually, it ends up being more complicated and interesting than you might think. So what really is a gene? This is how the talk will work today. Just to keep track of where we're at in the talk, we'll see across the bottom of the screen on some of the slides, we have a gene for us, a stretch of DNA, a stretch of RNA actually, and across the RNA, this little ribosome will move, just to show where we're at in the talk, and also to kind of illustrate the basic idea, which is the fundamentals of molecular biology that DNA is transcribed into RNA, which can then be translated into proteins. Over today's talk, we'll look at a few different topics. We'll start with the historical background of how people have thought about genes through history and how we tend to think about them today, but then some unusual examples that seem to break our usual definitions of what we might think a gene is. We'll look at some more detailed definitions that we could give of what a gene is. And then I'll conclude by looking at where I think this is going in terms of future research and where my research interests are for the future. I'll ask you a couple of questions through the talk, which you can respond to in polls or in open answers. And then there'll be time for some question and response at the end. So before I get into the topic of what is a gene, it might be interesting to see how I got to where I am researching this kind of topic at the Wellcome Sanger Institute. So how did I get here? In terms of my educational background, I went to school in New Zealand at a state school in Auckland. And then in university, I studied both biology and philosophy as an undergrad. So I did a bachelor's degree in both science and arts, and then moved into postgrad study focusing on molecular biology, focusing on things like genes. My PhD was specifically in an area called experimental evolution. So that's trying to look at evolution in the lab with studying this organism that we see at the top right. This is Saccharomyces cerevisiae, baker's yeast, the yeast that's used in baking bread, among other things. But studying it in terms of evolution, so studying in systems called chemostats, where the organism can grow and reproduce over many generations over the course of a few months. So over a few months, you can see hundreds of generations of yeast and track how evolution is happening before our eyes, as it were. So I was studying the data from this kind of experiment and doing some of these kinds of experiments. So after my PhD, which took about 4 years, then I moved to Germany for my first postdoc. Postdoc means after PhD, just postdoctoral. So that research project was for a few years, where I was studying bacterial genes, so switching from yeast to bacteria, and I was studying genes that overlap each other, which is an unusual concept of in terms of genes, and I'll explain that a little bit more in future in a few minutes. Then I had a short postdoc on a similar topic somewhere else in Germany, in Karlsruhe, and then I moved to the Sanger in order to kind of broaden my research to cover a variety of related topics around what genes are and how they evolve. Okay, so that was a quick introduction to me. Now let's think about the history of how people have thought about genes over time, because people have had all kinds of different ideas of what a gene is. Before we get into that, I want to ask you a question, thinking about trying to make it more tangible in terms of what gene is, how big it is, what it is. How many genes, how many human genes do you think would fit into 1 millimeter? So this is a poll that Sam will put up. And we have a few different options. So how many human genes would fit into 1 millimeter? Less than 1, about 1, or about 100, or maybe about 10,000. So I'm curious what you think.
Yeah, so I've just launched that poll, so hopefully that should pop up for everyone. So yeah, we'll just give you a minute or so just to put in your response. And then we'll go through what the answer is. Yeah, there's a few still to respond, so I'll give you sort of 30 seconds or so to fill in the response if you haven't done so already, and then we'll move on. Okay, I will end the poll now and share the results. So we have sort of a couple of people saying less than 1, similar amount saying 1, and then we've got the next highest was then 19% saying 100, and then 74% saying 10,000. Over to you, Zachary. What's the right answer?
Yeah, so 18 of you were right. It's actually 100. So they're not as small as most of you were thinking. Human genes are actually quite long. They're about 24,000 base pairs, 24,000 individual letters. Of course, we can represent that in a really tiny way or a really long way. We can write out the letters however we like. But as a chemical, as DNA, the physical size of a human gene would be about 1/100th of a millimeter. What are genes? In the kind of abstract sense of the units of heredity, they're the way that traits get passed down from one generation to the next. But that doesn't tell us very much about what they actually are. It just tells us what kind of function they fill, what they actually do, that they help to pass traits down. But that leaves open the question, what are genes? And it took a long time and a lot of science to work out what genes actually are. And this has of course changed as we've learned more over time. So there are lots of course, ancient ideas about how one trait was passed down through families over time. The Greek poet Homer attributed inherited properties of men, at least, only to their fathers. So he thought the information was somehow being passed from father to child, but he didn't seem to see a role, at least when the baby was a boy, for the mother. But the later Greek philosopher Aristotle didn't know that both parents contributed information to the child. So that was a development that he realized by, I guess, looking at families and thinking about how traits are being passed down. But he thought that things that you acquired over your lifetime were also passed down to your children. And there are some cases where that is true to a small extent, but in general, that's not the case. That in general, things that we acquire over time don't get passed down to the next generation. Over the next few centuries, people started to realize specific examples of traits that could actually be inherited versus being random or the product of the environment in some way. So one example of that was particularly striking was the blood disease hemophilia, and that is very clearly inherited. And that was discovered by the Muslim medieval surgeon in Spain, Al-Zahrawi, in around about the 10th century. Okay, so there were these kind of very broad ideas of heredity, of how things are passed down. But in the last 100 or so years, we've learned a lot more about the details of how this has happened, of how genes are passed on and what they actually are. So, of course, someone who's really famous in the history of genetics is the Austrian monk, Gregor Mendel. And he discovered through very detailed experiments with plants that genes really are discrete units, which went against the common thinking of the time that genes are somehow blended together over successive generations. So he learned that they are, at least in some cases, and in the simplest case, they are very discrete units that are associated with discrete traits, that they are kind of separable, they're separate. So that was a really important insight. A later really important insight that came just a few decades later was from a fruit fly researcher called Thomas Hunt Morgan. And he realized that the way that these genes are being carried down from one generation to the next is on chromosomes. And he discovered this in a really clever way by seeing that some traits are more closely linked to each other than other traits. So things like the color of the eye of the fruit fly, you can see how often this is associated with other traits. And if you have many fruit flies with lots and lots of children and many generations, you can start to work out how these traits are connected to each other. And it's actually a really clever insight that some traits are more closely connected than others, and that relates to their physical connection on the chromosome. Okay, so by this point, we've learned that genes are somehow involved in heredity, that in some sense they're discrete, they're separate units, and they're located on chromosomes. So you might think that we've kind of worked it all out by that point, but actually there's still a big question that remains at this point, which is, what are they actually made of? Are they made of DNA or are they made of proteins? Now, of course, we know that genes are made of DNA, but this wasn't obvious at all, because it turns out that chromosomes are actually mostly made of protein. So it was the natural thought that given that chromosomes are the mechanism of inheritance, and chromosomes are mostly made of protein, probably proteins are what make what's where the information is stored and what's making up the genes. Again, it was a really clever experiment that showed that it's actually DNA that's storing the information. And this was shown by infecting a bacteria with a virus and only passing on the DNA component of that virus to a bacteria and showing that that was enough to transmit the trait. So in other words, you don't need the protein in order to transmit the trait. The DNA is enough. That implies that DNA is the chemical that's really storing the information of genes. Still, lots of questions remain about genes after that, even though we've worked out what they are chemically. People didn't know what was the structure of DNA and how was that involved in it replicating or involved in being a gene. And this was discovered by a team of researchers, and some of the really important early experimental work was from Dr. Rosalind Franklin, who was producing X-ray images of different chemicals and interested in this question of how, what structure the DNA might have and how that might relate to how DNA is being replicated and then being involved as the basis of genetics. So this was discovered by her and other researchers in Cambridge in the late 1950s. And this was a really important insight, that there was this connection between the structure of DNA and how it was actually functioning. And this idea of the relationship between structure and function is something I'll touch on a little bit in the rest of the talk. So around this time, before we'd actually worked out some of the details of what genes are, there was this basic idea that one gene leads to one enzyme or one protein that has a particular function in the cell. This is a very intuitive idea, very simple, and it explains a lot of biology. Actually, it's really important for understanding how genes work. And this basic idea of one gene, one enzyme, once we got the molecular details of the structure of DNA and how it's related to RNA and protein, turned into what's often called the central dogma of molecular biology, that DNA leads to RNA, which leads to protein. That DNA stores the information and RNA is a messenger molecule that can transmit the information. So DNA is transcribed into RNA, which is then translated into the final functional unit in the cell, which is protein molecules. So this is a really simple, straightforward kind of flowchart of how information works in the cell, but it's stored in DNA, is moved in RNA, and its final functional form is proteins, which can do things that are useful for the cell. This is a really useful concept of a gene, and that tells us that genes are bits of DNA that get turned into proteins. But it doesn't tell us everything about genes, and it turns out there's some exceptions to this, which I'll go through. But proteins are really important. They do so many things in the cell, and the way they work together is really interesting, and there's a lot of details that we can learn there. So proteins are the basic molecule of life that performs most of the functional things that life does. They're arranged together into complexes. We have multiple different proteins together. That's a protein complex. They can be arranged into pathways where one set of proteins then interacts with another set of proteins, which interacts with another set of proteins to have a kind of a cascade of different events. They can be arranged into organelles that have kind of a set of complexes that are related together into some task, and machines as well that perform a particular function. One of the really nice machines in across cells, it's found across life, is ATP synthase, which is this molecule which is involved in energy production and transfer in the cell. And so this is one of the key machines of life, and it's a protein machine made up of different protein subunits. Each of these proteins is encoded by a bit of DNA, which is a gene. So this is the basic picture of life and explains a lot of molecular biology, but there are some exceptions, some unusual examples, which we'll go through. So one unusual example which was discovered very early on is that not every gene actually encodes for a protein. Sometimes this flowchart stops before it gets to proteins and we just have RNA. And that's enough to do some useful things in the cell, and is really important. So a gene doesn't need to code for a protein to be useful or functional. Many genes just get transcribed into RNA and can have a function just as RNA molecules. For instance, the core of the ribosome is composed of both RNAs and proteins, and they're both functional units that are important for the ribosome, the machine that's involved in translating RNA into protein. There's many examples of these RNAs that are not coding for proteins, but still have some kind of functional usage in the cell. They do something useful without needing to be converted into a full protein molecule. So that's one example, and there's lots of different details that could be discussed there. Another quite different example, which is a lot of my research has been about, is this concept of overlapping genes. I have here a picture of the COVID virus, or a kind of cartoon of the COVID virus, SARS-CoV-2, because it actually has a number of these overlapping genes, and I've studied some of them. So I've tried to illustrate this idea of an overlapping gene here, that one bit of DNA can actually produce one RNA that can produce multiple proteins, or it can produce multiple RNAs that can produce multiple proteins. And there's different ways that this can happen, but the simplest one, which I'll show here, is an example from SARS-CoV-2, the COVID virus, where the same bit of DNA, these letters here it's RNA actually, sorry, AAUGGAC. This is the basic RNA string, which in most organisms would have been converted from DNA. In SARS-CoV-2, it's actually transmitted just as RNA. It doesn't initially start as DNA. So we have this nucleotide this set of letters, which encodes a protein, the nucleocapsid protein in blue. This is straightforward. This is the standard, more or less, the standard picture of the gene. The complexity is that the same string of letters can actually also encode a different protein if we shift the letters that we're reading by one. So instead of starting with AU here, we start with AUG, and we read a completely different protein, which has a completely different set of amino acids here and a completely different structure. So one gene, or one segment of DNA, or in this case RNA, can actually encode for multiple proteins. So this also undermines the kind of standard dogma that one DNA gene leads to one RNA gene, which leads to one protein. So those were just two kind of counterexamples to the kind of central dogma, the basic idea of molecular biology. And there are others as well that we could talk about. But I'm particularly interested in how this concept of molecular biology and different kinds of genes and exceptions to the standard view of genes relates to evolution. Proteins have different histories. There's lots of different proteins in cells. We can compare their histories to each other. Some proteins have very ancient ancestors, so you can trace their family tree all the way back to somewhere near the origin of life. And others have evolved very recently. They're only found in a small group of species. They've only evolved over the last few million years. And proteins are have different histories, some very young, some very old, some in the middle. But the newly evolved proteins have quite often some quite unusual properties, and I'm really interested in these, because these are also kind of on the boundary of what we say is a gene and what isn't a gene. If they have unusual properties, it's actually really hard to classify them. So proteins which have evolved recently often have unusual proteins when we compare them to standard proteins that we might think of, which are often very ancient. The genes encoding recently evolved proteins are often short. They're often only weakly expressed. So there's not many copies of them in the cell. There's not thousands and thousands of copies. There's only maybe 1 or 10 or a few. And they have various other properties where they're not as well structured sometimes as older proteins. They're not as complex. So if you look at them, they don't look like a standard gene. And that's because they've evolved recently and they haven't had as much evolutionary processes acting on them, producing the complex genes that we're more familiar with. Okay, so those were some really quick examples of what a gene can look like and some kind of counterexamples to the usual picture of what a gene is. And this raises the question, well, what really is a gene? How can we define what is or isn't a gene, given there's so many examples and counterexamples. In order to start thinking about this, we have another poll, which I'll let Sam run. So what is the function of an object? When we say a gene is functional, that it does something, what are we really saying? Are we saying that the function of an object is what it does or what it interacts with? That's option A. Or are we saying the function of an object is what it was selected for, what it was designed or intended to do? That's option B. Option C is how the object contributes to a larger system. Or option D is something else. Which of these is the best way when we have an object in front of us, we want to know what its function is? How do we work that out?
So the poll is live. So we'll just give everybody a minute or so to put in their response. Thank you as well to those that are putting in questions in the Q&A section. That's great. Do keep putting any questions that come to mind there, and we'll get to them when we get to the Q&A bit of the talk. Lots of people putting in their responses so far. I'll give you 30 seconds. We'll go 30 seconds so that everybody gets the chance to fill a response if they would like to. I can see, looking at the responses live, that it's a little bit of a toss-up between the first three. So we'll have a look and see what it ends on shortly. Okay, 10 seconds or so. So if you haven't put in your response, do so now, and then we'll find out what is the function of an object. Okay, I am going to end the poll now and share the results. So we have 36% saying what it does or interacts with, 37% saying what it was selected or designed to do, 24% saying how it contributes to a larger system, and 3% saying something else. So back to you, Zachary.
Yeah, great. So this was a really hard question, and there isn't a single right answer to this. Philosophers have debated it. There's a large literature what it means for something to be functional, and specifically we can apply this to a gene. What's it mean for a gene to be functional? And there's some very kind of heated debates actually in the literature and about what it means to say that something is functional. And my takeaway from it is that it's actually a mix of all of these, and it will depend on the context of what we're trying to look at as to what counts as functional. I'll go through each of these a little bit to think about it, but I'll go through it by thinking about a specific example of a set of genes in bacteria, the kind of thing that I'm interested in looking at. So this is a transporter system in bacteria, seen in different ways from a paper from a few years ago. And this helps to transport molecules across a membrane, basically. And it's just a really nice image of a bunch of gene products, so a bunch of proteins that are interacting with each other for a function. And the function is transporting molecules across a membrane. And there's a lot of kind of science that's gone into making these images and working out what the overall structure looks like and how it functions. Okay, so what's it mean to say that a gene in this system is functional? The first option is that the function of a gene is what it does, its causal role, the way it causally interacts, what it causes, what it interacts with in the cell. So on this definition, a gene is functional if it does something biochemically in the cell. If you can do a biochemical measurement associated with the gene, then you can say that's what it does, and therefore the gene is functional. So for instance, in this case, we have two proteins, or actually sets of proteins, say the green and the gray. If they're binding together, if they're interacting with each other, we could say that's a function of the proteins, and therefore that's a function of the gene that encodes the proteins. So that's one definition and one way of thinking about the function of a gene. But then there's a very different way, which a lot of biologists will have, which is how the gene got there will contribute to what we think the function of the gene is. So in this definition, a gene is functional if it was subject to natural selection in the past for a particular function. So for instance, for our protein, our transporter system, if the function of the proteins is their role in transport across, the function of the proteins is their role in the transport of a molecule across the membrane, because that function gave the ancestors of the bacteria a growth advantage, that this contributed to their fitness, to their flourishing as bacteria, and therefore these genes and these proteins have this function because it helped the bacteria in the past. If it helped the bacteria in the past, then it was subject to natural selection, it was selected for, and therefore the protein complex is functional. And this is maybe the standard definition of function that at least evolutionary biologists would have, whereas the first definition is maybe more what biochemists would tend to think about. So that's the second definition. A third definition, which is also separate, is how does this gene contribute to the wider system? How does it contribute to the organism? And this is the physiological definition. How does it contribute to the physiology, the system of the organism? So in this description, each gene plays a role in the wider system of a cell, usually as part of a subsystem, such as the transporter complex. It's more complicated in multicellular organisms because then you have multiple cells, which can then also have functions within tissues or within organs. But the idea here is that how you tell whether something has a function is whether it's playing a role in a wider system. That's a separate question from how it got there, the evolutionary history. And it's a separate question of whether it just interacts with something, it has some causal or biochemical effect. So here's three quite different definitions that are all used by biologists in thinking about the function of a gene or whether a gene is functional, just to remind you that we're thinking about this protein and transporter complex. So we can study each of these definitions with different methods. We can study what a gene does with different biochemical techniques. And there's probably hundreds of biochemical techniques we could use to study what a gene does. We can study how it got here, and this is largely what I do as an evolutionary biologist, by comparing genome sequences to each other and trying to work out what their history was. But we can also study how the gene contributes to the overall cell or the overall system of the organism with systems biology, and thinking of how the subcomplexes and complexes are interacting with each other. And like a lot of biologists, I use a little bit from each of these different approaches in thinking about what a gene does, what its function is. Okay, so that was quickly exploring these different definitions of what a gene is, whether it's functional, what it does in the cell. What does this mean for us moving forward? Again, we have a quick question for you to see what do you think about this? Given that we actually don't fully understand the genes of any organism, that we don't know with complete certainty how many genes there are in the human genome, we don't even know how many genes there are in SARS-CoV-2, the virus that causes COVID, or in very well-studied bacteria. There's a lot of dubious cases we're not sure about. Given this, which organism or virus, aside from humans, do you think it would be most useful for us to fully understand genetically, to know what all the genes are and what they do? Which organism should we focus on?
So this is sort of an open text question, so you can type in responses, hopefully, to this question. So again, we'll give people a minute or so to fill in a response on this one. So again, if you weren't sure on the question or just missed that, it's which organism or virus aside from humans do you think it would be most useful for us to fully understand genetically? And you should see a little box where you can type in a response if you're able to. So we'll give people 30 seconds or so to fill in a response. Thank you to those that have already submitted an answer. That's great. Okay, I will say 10 seconds or so. So if you haven't put in your response and you would like to, please do so now before I press end poll. Okay, I am going to end the poll now, and we will share some of the results. So hopefully, Zachary, you can see some of the different answers that are coming up. Don't know if you can see. We've got so we've got mouse and different bits of that. Let me stop sharing and I can go through some of them actually in a second. I'll read some of them out for you. So we've got I think this is meant to say bacteriophages, C. elegans. We've got someone saying mouse, the flu virus, HIV. What other ones have we got? We've got HIV come up a few times, chimpanzees, fruit flies again, viral genome, insects, mice, wheat, quite a few fruit flies as well in some of the responses. Yeah, E. coli as well. So yeah, quite a few responses there.
Yeah, great. And again, of course, there's no right answers, and they're all useful to study. What's interesting is most of these were model organisms, which I think is a really good approach. So these are kind of standard organisms. There's only a few of them, like E. coli and yeast and fruit flies and mice, that have been studied a lot. So these are good starting points because we already know a lot of background about them, about what the genes might do. So it's a lot easier to add knowledge into a system where you already know a lot of the background. And that means new knowledge is also really useful because we can relate it to what we already know. So that's a good and useful approach that science has tended to use when thinking about how to understand the genes of different organisms. So yeah, like I said, we still don't know exactly how many genes there are in humans. There's actually some ongoing research in that, and I have a small project related to that as well, trying to study some of the cases that are unclear whether they really are genes or not. We don't even know in SARS-CoV-2, which I think is really surprising given how many people have looked at that, but there's still some edge cases which are controversial or unclear whether they really are genes or not. And again, it's going to depend on what you count as a gene. The best studied bacteria is E. coli K12, and even there where there's some edge cases that are not clear whether they are genes or not. The answer will depend on our definition of a gene. I think a lot of people don't realize that fully, so that doesn't help in the discussions about it. But if we're clear about what our definition is, then we can also start to be clear on how many genes we have and which ones are functional. Because we don't know how many genes there are, and we certainly don't know what they do fully, that means we don't even understand the parts list of some of the best studied organisms, the individual protein components. We're still not sure how many there are, which ones are functional. Once we have that, then there's a lot more to learn about what they actually do, how they interact under different conditions. So there's a lot more research to do in genetics, even for these very well-studied model organisms, as well as all the mini organisms, which we know even less about. I would say that studying unusual genes, the kind of research that I'm doing, will help in understanding lots of different things. Basic biology, just how organisms work, more applied questions that we're very interested in, like how does cell division go wrong in cancer? Some of these unusual genes are actually involved in different cancers. There's a lot of research on that. How organisms become harmful in infectious diseases, because there's many different infectious diseases and different organisms have different genes. There's a lot more to study there. And also the basic question of how did we evolve and how does evolution work? There's a lot that can be learned about that by studying these unusual genes. My own research is focusing on how newly evolved genes that have quite recently evolved contribute to cells, the cellular system and bacteria, and what it tells us about evolution in general. Eventually, I'm hoping it's going to be applied in terms of antibacterial treatments as we understand these bacterial systems better. Okay, so that was my summary of some of the history and ideas around what a gene is. And I look forward to your questions from what I've brought up.
Amazing, thank you. And we've already got quite a few questions in the Q&A section, so I think we'll just start going through some of those. So, but if you have any questions after that, do put them in that Q&A button that you'll see at the bottom. But yeah, so the first question that we've got come in is, how do you identify where a gene starts? Same base sequence for all.
Yeah, so that has a simple answer, which is usually it starts with ATG, and then it has a more complex answer in that it can actually start with many other codons. It can start with CTG or TTG, but in some bacteria, occasionally it will start with other things as well. So again, there's kind of borderline cases, and we still don't fully understand that part either. But usually it will start with an ATG set of nucleotides.
Amazing. Okay, so moving on to the next one, it says, what percent of the 20,000 human exons do we know the function of?
Yeah, so unfortunately, I don't really focus on human genetics. I don't know that much about it. But I'll just say that there is a large proportion that we don't know. And there's also a number of these very short genes that we're not sure how many of them there even are, let alone what they do. So I can't give a proportion of that for humans. For bacteria, even in quite well-studied bacteria, there's about 10% of genes we don't know what they do. In the less well-studied bacteria, it can be 50% of the genes we don't know what they do. So that varies. Humans, there's a lot of focus on, so it's probably smaller, but there will be a few percent that we're not sure what they do.
Cool, amazing. So the next one says, how many times is mRNA translated as at a polysome? Is it 50 times at the telomere is reduced by one base at each translation? I don't fully understand that statement, so hopefully, Zachary, you might be able to unpick that one a bit for
Yeah,
me.
so I saw that question. I think the answer is just once, if I understood the question, that the ribosome just goes through once, and then if it's going to, and that makes one protein, but then it can go back again and make another protein. So every kind of trip down the gene, every traversing of the gene by the ribosome equals one protein. And there's lots of proteins in the cell, many thousands, I think, and they're all kind of translating genes constantly. Yeah.
Cool. There's loads of questions coming in, so I will try my best to get through them as best we can. So the next one that I'm seeing is, say, can we call products of overlapping genes as isoforms?
Yeah, that's an interesting question. I don't think so. So the usual way that isoform is used about proteins is if you have a gene, but it has a different start site. I should have actually mentioned that as another example. So you can start the gene here, but you could also start it further downstream and make a different version of the protein. That's called a protein isoform or protein proteoform. An overlapping gene is a completely different protein sequence. So it's actually a bigger change than an isoform would be.
Okay, amazing. We've then got one that says, do most proteins have more than one gene coding for them in the DNA, i.e. the same protein on different chromosomes, for example?
I thought that question was going in one direction and then it kind of branched in a different way. So I'm not sure. Can you repeat it? Sorry.
Yeah, so do most proteins have more than one gene coding for them in the DNA, i.e. the same protein on different chromosomes, for example?
Okay, I guess that's the question about human genes. That's not that common. I don't know how common that is. But it also depends on what you count as the same protein. There are protein families that diverged a long time ago, millions of years ago, and like different forms of hemoglobin. I was just watching a lecture about that. There's lots of different copies of that, and they'll be on different chromosomes. But usually, there's just one location in which a protein is translated from.
And this is a sort of from the same person, but has asked, which might link in, I think, actually, which is how many organisms have the overlapping genes that you described? Is this common across organisms?
Yeah, so we don't know, but there are examples in humans and I think in mice and fruit flies and yeast and bacteria and viruses. People used to think it was just in viruses because they have very small genomes, so they need to compress the information that they store. But we've discovered that there are actually quite a few of them in bacteria, in archaea, which are kind of similar single-celled organisms, which are quite different to, well, in some ways similar, in some ways very different to bacteria. So they're everywhere, and we don't really know how many there are. But they're also very probably particularly common in viruses.
Okay, cool. So next one, as I say, I'm trying to make sure we can get through all of these, but yeah, thank you for putting in all of your questions. So it says, how does the dominant allele in the heterozygote prevent expression of the recessive allele?
So yeah, that's a question more for probably a human geneticist who's
Okay.
with diploid organisms. I'm going to skip that question.
Okay. Yeah,
Maybe
let's we can
and see what it tells you. But yeah,
we can always, if there's any questions that we can't, we can always see if we can get the answers to you as well. I'm going to just quickly move down as well to see some different. So do overlapping genes occur in forward/reverse strand, or can it be overlapping in the same direction?
yeah, that's a great question. So the example I showed was actually in the same direction, but just shifted by one. So that is maybe the most common, but you can also get forward and reverse strands. And most of my research has actually been the genes that are on opposite strands. So one is on the what we call the plus strand, and one is on the minus strand, just because those are easier to study. But there's also many cases where they're just shifted by one or by two. So yeah, all of the possible combinations can be found.
Amazing. Okay, so this one is a little bit about I guess the future looking. So this one is saying, looking ahead, in which areas do you foresee the field of genetics focusing, and what breakthroughs do you anticipate scientists will achieve in the next decade? So I guess that's quite a broad question potentially. Maybe just specific on some of those points that maybe in your area, where do you see some of these things that you were touching upon maybe moving to?
Yeah, massive question, lots to say. But one thing is that sequencing is now quite cheap. So we have all this genome data, but the real bottleneck or the real difficult step is interpreting the data now. So this is why we need more people trained in genomics and bioinformatics to really try and make sense of the data, because making the data is quite cheap compared to how much it used to be. But interpreting the data is much more difficult. So that's where a lot of the advances will be in terms of using the data that we already have and really making the most out of it. And maybe the big thing that will happen over the next few years is like personalized medicine, where people can have the individual genome sequence, but also the genomes of infectious diseases that are affecting them, or cancers that are affecting them. So I think because sequencing is becoming affordable, we can go in much more detail for individual people to respond to the medical problems that are related to genetics. So yeah, that will be a massive field in the next 10, 20, 30 years.
And this might link into this other question that we got, which was a little bit about how can computational biology help aid gene identification across the genome?
Yeah, so I guess this is my field of research, and one of the main ways is comparing genomes to each other. So we see if two organisms have the same bit of DNA that has a certain pattern, because DNA changes, but in genes, the changes will be different to outside of genes, because if you want to retain the same function, you're going to be limited in how much you can change it. So that will show up as a different kind of pattern than two bits of DNA which aren't doing something useful or functional. So that's one of the main ways that computational biology is useful, is we now have massive data sets in bacteria of potentially hundreds of thousands of genomes we can compare to each other to really see how the DNA is changing, and which bits look like they're functional encoding for proteins, and which bits look like they're functional encoding for RNAs, and what the difference are.
And this might not this hasn't been answered but asked but I guess in terms of computational biology and the use of, you know, we're hearing more about like AI and machine learning. Is that in this type of work? Is that something
Yes.
that's you're looking at?
Yeah, because the patterns of differences, for instance, are really complicated. And you can do kind of basic statistics, but when you have hundreds of thousands of genomes, these artificial intelligence methods become really useful. And you can, if you have a set of genes that you can train on, you can learn the kind of patterns that are associated with genes that we might not spot as humans, but artificial intelligence models can spot some of these patterns that we might not expect and use them to make more accurate predictions and discover more about the functions of genes. So yeah, this is a massive field.
Amazing. There's going back to sort of basics really, which is, I guess, what's the difference between genes and chromosomes?
Yeah, okay, yeah. So I should have explained that better. So the chromosome is the full, well, it's a single molecule of DNA. Some genomes will just have one chromosome, like a typical bacterial genome will just have one circular chromosome. Some genomes, like the human genome, will have multiple chromosomes. But it's the full stretch of DNA, whereas the gene is just a short segment of that DNA, which is then doing something useful, whether that's encoding an RNA, which might do something useful, or also encoding a protein molecule. So yeah, the gene is just a short segment, usually maybe a few hundred or a few thousand letters, whereas the chromosome can potentially be millions and millions of letters of DNA long.
Amazing. Okay. Just going through some of the other ones, and apologies if we don't have time to go through all of the questions. It's been, yeah, thank you for putting them in. We can always see about sort of we can always try and provide sort of places to go to try and answer some of these as well. So how useful are newly evolving genes in natural selection?
Yeah, so that's a good question. In that there's very little that we know about these newly evolving genes, and it's a big question whether they are under selection at all. And there's kind of conflicting evidence on that. But there is some evidence that at least some of these are under natural selection, even though they've just recently evolved. And those are the ones that might be particularly interesting. But there's a lot that don't look like they are under natural selection, but they still do something in the cell. And that's a bit mysterious. That's why these different definitions of what a gene is really important. That sometimes with different definitions, you see different genes. And in this case, just because something isn't under natural selection actually doesn't mean that it doesn't do something in the cell. So yeah, they're quite separate questions that can be kind of explored in lots of detail. And the answers we're still working out the general rules of this kind of thing.
And do you ever see there being a general consensus on that definition happening?
It's probably going to be different because biochemists are going to be interested in different things to evolutionary biologists. So at some point there's going to be some separate, but there will need to be standard definitions for when we have our official lists of genes, and that's what people are kind of working on now.
So you were saying about sort of your work on bacteria. There's a question that sort of says that are you working with one specific bacteria, or are you focusing on sort of on all or a broad range?
Yeah, so most of my work is in E. coli, which is a standard model organism. But because I'm an evolutionary biologist, I'm comparing it to different organisms. So I'm comparing it to different strains, so different versions of the species. I'm comparing it to different species, closely related species that are in the same genus or the same family. And some of my work is looking across all of bacteria for what are the general features of these recently evolved genes. But most of my work is specifically focused on E. coli, because that's the area that we already know quite a lot about. So we have a lot of background knowledge for.
Okay, amazing. And I think we'll just end on one last question, just given the time, which might be quite a nice one to wrap on, but which is basically what determines the evolution of genes?
Oh, that's a really big question that I guess if we understood that, then there'd be a lot less research to do. But so yeah, we don't, there's a lot of things about evolution that we don't have kind of general principles for. We just see individual cases, which we can study, but we don't understand the broader kind of laws, if there are laws of evolution. So one of the things that determines evolution of genes is functionality, like what does it do? And in general, if something is really functionally important, then it's more constrained and it will evolve slower. But it becomes really complicated. There's lots of exceptions to that. That's why evolution is really interesting, because things evolve differently depending on the organism and what they do. So yeah, there's not really general rules, but we're starting to get some hint at some of these principles, which we can start to apply.
Okay, amazing. Well, we're just noticing the time, so I just want to sort of bring it all to a close. So I just want to say thank you to Zachary for joining us this afternoon, this evening. Thank you to everybody who has come along and joined us to watch the event. If this was your first Genomics Lite and you really enjoyed it, I'm pleased to share. If I share this link in the chat, this will take you to the page where you can sign up to next month's event. We are looking at gene editing. So we've got the head of gene editing coming to join us, and we will be taking a whole look at that field, which is again something that is massive. So do come along. But yeah, once again, thank you ever so much, Zachary, for joining us. Thank you for all of the great questions. Sorry if we didn't manage to get through all of them. I will look through them after the event and make sure that we can sort of go and through that. Someone has just said that they can't find the link. I might have accidentally just sent that to myself, so let me post that again. Hopefully that goes through. Give me a shout if not. But yeah, thank you for joining, and I hope everybody has a lovely rest of your evening. And yeah, it's been lovely to have you with us. I will stay on just for a little bit longer because I know that there was a few messages just about specific things that I will reply in terms of recordings, but this talk will be recorded, has been recorded, and it will be posted onto YouTube and our website, so you will be able to watch this back as well. So thank you.